Welcome to the Induced Innate Immune Responses module. And in this talk, we're going to learn about the extravasation of immune cells. We're going to first look at chemokines and different groups of chemokines, and then learn about the different events that occur during the extravasation of monocytes and neutrophils. Inflammation normally occurs when the host is able to recognize pathogens that are present inside. The role of inflammation is to direct molecules as well as cells to the site of infection, and these molecules and cells will then play an important role in clearing the pathogen. Another role of inflammation is to activate the immune cells so that they do a better job at clearing the pathogen. Inflammation also induces localized blood clotting, and this helps in curbing the spread of the pathogen to other parts of the body. And finally, inflammation also helps in repairing injured host tissues. Now, inflammation is triggered by the production of different molecules like cytokines, chemokines, and other types of mediators like histamine. Now let us look a little more in depth into the role of chemokines and how they promote chemotaxis. Now chemokines are made in the earliest phase of infection, and they induce directed chemotaxis, and the result of this directed chemotaxis is the movement of cells towards the source of the chemokine. Now there are different groups of chemokines, and we're going to look at 4 such groups. The first one are called as the C chemokines because they have a cysteine, 2 cysteines that form a disulfide bridge. Another group of chemokines are the CC chemokines because, as you can see in the figure in the black box, there are 2 cysteines that form 2 disulfide bridges, and these 2 cysteines are right next to one another in the primary sequence of the chemokine. An example of another chemokine is the CXC chemokine family. Now in this case, as we can see in the black box, there are 2 cysteine amino acids, but there is another amino acid that is between them. Hence the sequence is a cysteine, another amino acid, followed by another cysteine. And the 2 cysteine once again form disulfide bridges. The last one is going to be the CX3C. In the case of the CX3C, if you're following the pattern, then you can already guess that there are 2 cysteines, but between those 2 cysteines, there are 3 amino acids. And thus the sequence is a cysteine followed by 1 amino acid, 2 amino acids, 3 amino acids, and then another cysteine. Chemokines are able to recruit immune cells to the site of infection, and in the following slides, we will look at the mechanism by which this occurs. Now, immune cells need to leave the blood vessels so that they can come to the site of infection. So the monocytes and neutrophils that are part of the innate immune system are usually circulating in the blood. However, when they have to go to a tissue where there is an infection, they have to leave the blood, and for that process is called as extravasation. Now, in order for the cells to know where they're supposed to go to or where that site of infection is, that is where chemokines play a role. So chemokines help the immune cells to leave the blood vessels so that they can go to the site of infection. So let us see how this process actually works. For leukocytes to leave blood vessels, they must be able to interact with the endothelial cells that are present in the blood vessels or that make up the blood vessels. So this interaction requires protein-protein interactions, and the proteins that play a role in interactions between the endothelial cells and the immune cells are called adhesion molecules. The different types of adhesion molecules that are involved in the extravasation of leukocytes are, the first one is the selectins. And the selectins that we're going to look at is the P-selectin and the E-selectin. And they are expressed in endothelial cells. Another adhesion molecule that we will look at are the integrins. The integrins in this situation are present in the innate immune cells. Another set of proteins that play a role in extravasation are the immunoglobulin superfamily, for example, the ICAM. Now, ICAM can be present on both endothelial cells as well as activated immune cells. Let us look at the process of extravasation now. Now, we all learned that when there is a pathogen present, it can activate the complement pathway, resulting in the production of anaphylatoxins like C5a. We also learned that our own immune cells and epithelial cells can also produce pro-inflammatory cytokines like TNF-alpha in response to the presence of a PAMP. These two molecules can activate endothelial cells. And let's look at an endothelial cell in the figure. And if we focus on the figure now, in the endothelial cells, there are granules present. And these granules have the protein P-selectin. The presence of the molecules like C5a and TNF-alpha induce the externalization of these granules, and that results in P-selectin being present on the endothelial surface. Once P-selectin is present on the endothelial surface, it can interact with the leukocytes, and we will see how that happens in the next few slides. What should be noted is that P-selectin is the first selectin that is expressed on the endothelial cells during an infection. Once P-selectin is expressed on the cell surface, there is a signal in the cell to induce the expression of another selectin, which is E-selectin. And once E-selectin is expressed, it will also be transported to the cell surface. In this slide, let's focus on the figure. So in the figure in the black box, we can see the endothelial cells that are expressing E-selectin on the side that is facing the blood. So the part on the black box right now is where the blood is flowing, and in the blood we will have our blood cells, including monocytes and neutrophils. The presence of E-selectin on the endothelial cell is going to trigger a process called as rolling, and let us see how that happens. So let us focus on this neutrophil and look at its cell surface. And at the cell surface, we see a carbohydrate moiety that's called as the sulfated sialyl Lewis X. The presence of the sulfated sialyl Lewis X on the surface of the leukocytes like neutrophils and monocytes enables the cell to interact very weakly with P-selectins and E-selectins. Now, ideally, we would have the blood cells simply whizzing through the blood vessel, so they travel very fast. However, when E-selectins are expressed on the endothelial cells, the presence of these E-selectins, or P-selectins for that matter, will facilitate an interaction with the sulfated sialyl Lewis X present on the neutrophils, and hence the neutrophils and monocytes will start associating with the endothelium in a very weak fashion. This slows them down, and what we see is a process called as rolling. So instead of passing through the blood vessel very quickly, the leukocytes, like the neutrophils and the monocytes, are going to show rolling. After the step of rolling comes the step of tight binding or margination. Before we look into the step of tight binding, let us first focus on the chemokines that are secreted. Now, chemokines are secreted at the site of infection, and because they are soluble proteins, they can diffuse in the surrounding area as well as into the blood vessels. Once they cross the endothelial barrier and are able to reach the blood vessels on the side where the blood is, the chemokines can bind to proteoglycans that are associated with the endothelial cells. If you remember from cell biology, proteoglycans are proteins that form the extracellular matrix. Now, from our previous slide, we saw that the neutrophils have slowed down in their speed and they're rolling across the blood vessel because of the association of the selectin that is present on the blood vessel with the sialyl Lewis X carbohydrate moiety that is present on the surface of the neutrophil. Now, binding of the chemokine to the chemokine receptor can happen because of the slow movement of the neutrophil in this example. Now, even though I say neutrophil all the time, the same thing happens with monocytes as well. So going back to the figure, we can now see that that slow-moving neutrophil is now able to associate with the cytokine, or in this aspect, the chemokine, that is present on the proteoglycans because it expresses the chemokine receptor. This association triggers a change in the conformation of the integrins that are present on the neutrophil. The integrins can now interact with an ICAM adhesion molecule that is present on the endothelial cells, and that is what is shown in the black box. The interaction between the integrin of the neutrophil and the ICAM of the endothelial cell is a very strong interaction, and hence we see tight binding between the neutrophil, or a tight association between the neutrophil and the endothelial cell. This allows the monocytes and neutrophils to attach firmly to the endothelial cells. The next step of extravasation is called diapedesis. In this step, the monocytes and neutrophils are able to cross the endothelial barrier through the endothelial junctions. And this is what is shown in the figure in the black box, where we can see an immune cell squeezing through the junctions between two endothelial cells. This is facilitated with the help of integrins as well as other molecules called PCAMs. Now, the PCAM is present in immune cells as well as the intercellular junction of the endothelial cells, and these interactions of integrins and PCAMs allow the monocytes and neutrophils to squeeze between the endothelial cells. Once the immune cells are able to get access to the other side of the blood vessel, which is where the tissues are and most likely the site of infection is, the immune cells have to break through the basement membrane. And the way they do that is they secrete, or they make, extracellular matrix proteases that are able to break down that extracellular matrix and can enter into the tissues. The final step is migration, where the chemokines produced by the cells, they form a concentration gradient. And so the monocytes and neutrophils follow that concentration gradient because they express receptors for the chemokines and now are able to go to the site of infection. During an infection process, neutrophils are the first cells that will go to the site of infection, and they rapidly migrate to the site of infection in very large numbers. Monocytes also migrate, but however, they migrate a little more slowly, and you don't see them as rapidly as you see neutrophils present, simply because neutrophils are a lot more in number when it comes to how many cells are there in blood.
With this, we come to the end of our talk, where we learned about different groups of chemokines and functions of chemokines. We also learned about the different steps that are involved during the extravasation of monocytes and neutrophils, which include the processes of rolling, tight binding, which is also called as margination sometimes, diapedesis, and migration.